Welcome, everyone. As always, it's very nice to see familiar faces and new names. So, welcome to all new nano educators and nano enthusiasts. While I go over some housekeeping information, please introduce yourselves in the chat. Name, school, state, favorite science fact, anything else you would like to share with the group. Remember, this is a very informal setting, and we want to have a nice discussion. If you're new here, hello, my name is Maria Fernanda Campa, and I'm a staff scientist at the National Nanotechnology Coordination Office. Since 2022, my office, in collaboration with Marielle Kolker, a physics and engineering teacher at Morrison Town High School in New Jersey, and Danielle Duran, the Education and Outreach Program Manager at Nano at Stanford, who is also back teaching high school this year, we have created this space for teachers, nano educators, and nano enthusiasts to informally meet around 4 times a year to discuss different topics of interest. Past sessions have included nano and agriculture, nano and sustainability, lessons plans developed and implemented by middle school and high school teachers, etc. If you are interested in seeing past sessions, all of our sessions are recorded and posted on YouTube and nano.gov. I will share the link in the chat shortly. We also have a live Google document with links and resources shared from all past sessions. So if you see anything in the chat or anyone mentions something, I will make sure to capture that and add it to the live Google document. As a reminder, these sessions are driven by your interest. So please let us know in the chat or over email what would you like to learn about? What would you like to discuss? If you're interested in sharing how you are teaching nanotechnology in your classroom, please come share that with the group. We're always interested to see how you're introducing nano in your classroom, and it's also a great learning opportunity for all of us. Now, without further ado, I would like to welcome our special speakers for today. Today, we're welcoming Dr. Maggie Bilksley, a postdoc from the Hammond Lab at MIT, who will give us a crash course on nanomedicine, followed by her colleague, Christine Pace, the Education Outreach Coordinator at the Koch Institute at MIT and a former school teacher herself, who will discuss how to make this contest accessible to students. And with that, oh, I didn't even change my slides. But with that, Christine and Margaret, please take it away.
Thank you for the introduction. Let me just get these slides shared. Awesome. Thank you so much. Hopefully you're seeing slides now.
Yes, we
Perfect.
are.
Great. Awesome. Thank you for that introduction. I'll go and introduce, I guess, myself again, but I'm Maggie Billingsley. I have a PhD in bioengineering, and I'm a current researcher in the Hammond Lab at MIT. I work within the Marble Center for Cancer Nanomedicine, so kind of a conglomerate of a lot of us working on nanoparticle-related research. I myself have worked on gold, lipid, and polymer-based nanoparticles, specifically for cancer applications, so looking at cancer detection and cancer therapy. I've worked a lot with breast, ovarian, and blood cancers. And I have my email here. Happy to answer questions, of course, in our discussion today, but afterwards as well if any additional questions come up. And I'll kind of dive in just to a broad intro on nanomedicine. So I wanted to start off with giving a definition, so kind of what we're talking about today. And really, the definition of nanomedicine is the science of diagnosing, treating, and preventing disease, relieving pain, and improving human health using nanoscale tools. I also like this one, kind of the idea of setting the goal of nanomedicine, which is to treat the human body at the atomic and molecular levels. And I really like that definition because the way I like to describe nanomedicine is developing really small tools that'll have big therapeutic effects. So moving away from designing things like surgical interventions or kind of oral formulations, things like pills that we take, and diving further down that size scale toward nano, so we can make these really small tools. But when we say small, I feel like an easy question, of course, is how small is small, kind of defining that scale. So, of course, as the name implies, we're on that nanoscale in nanometers between 1 to 100 nanometers, which is something around a nucleic acid, protein, or a virus. So, something like an antibody, kind of in that range. So, something that is bigger than a molecule like water, but smaller than something like a bacteria or a cell. And nanomaterials and nanoparticles can come in a lot of shapes and formats, so they really range along that kind of nano of like nucleotide to virus. But if we think in a bit of a broader biological scale within the context of, say, like a cancer patient, we can think of a patient as being on the meters scale, and they'll often have tumors that are around a centimeter to millimeter scale. and the cells that make up that tumor are microns in size. And then we have our nanomaterials that are even smaller than that. But I myself find it challenging to really think through that range of sizes, especially when we go down to things we can't see. So, if instead we were to think about this biological size scale, instead of in nanometers, in micrometers, so instead about 100 millimeters in size, we'd have a nanoparticle, or nanoparticle would be about the size of a baseball if it were 100 millimeters. And then our cells would be, kind of by comparison, about the size of a baseball diamond. And then the tumor they're making up would be about the size of a city like Boston. And then the patient would actually be about the size of the planet Earth. So, really, we're designing baseballs that we're tossing at something the size of planet Earth, hoping that we reach the city of Boston and impact a particular baseball diamond. And so, when you say it that way, it sounds a little bit confusing as to why we would want to design something so small when our targets are so big, comparatively, of course. And so, I wanted to discuss kind of briefly why nano is so helpful for medicine. And really, the rationale behind this is that we want to get small enough that we can navigate biology on its own scale. So, as I mentioned, nucleic acids and antibodies and proteins are at this nano level. So, we also want to be designing therapies that are interacting in that same size category. And it's so we can use these nanoparticles to improve our understanding of the biology of disease and to better deliver medicines to ultimately improve therapeutic outcomes. So, kind of sticking with this baseball analogy, the size of something like a cell receptor or something we'd want to target on a cancer would be about the size of a baseball glove. And so, for our nanoparticles that are about the size of the baseball, you can see this is a good fit. We could really use a baseball to understand how that mitt works, if it's functional, and understand its use. And this would also work for something the size of a tennis ball or a softball, but if we go too big or too small, we suddenly can't really evaluate our baseball glove anymore. If we were to use a marble to try to deliver to a baseball glove, it'd be really hard to grasp it. We'd struggle to understand the function of the glove. And similarly, if we were to use a football or a basketball, something too large, we'd kind of fall into this same trap of not really exploring the uses of that glove or understanding kind of how to interact with it. So we wouldn't be able to utilize or kind of have a connection size wise. And so that's why, from a biological perspective, nano is really the right call. It lets us kind of communicate biologically with relevant features. But from a materials perspective, nano is also really helpful because of the idea of emergent properties. And so emergent properties can be physical, chemical, or even biological. But they're really driven by the idea that nanomaterials have a really high surface area to volume ratio. And surfaces, especially in material science, have a lot of really cool, kind of unique characteristics. There's a lot of studies of interfaces and edges on traditional bulk materials. And the way I kind of conceptualize this is if we think about a loaf of bread, the surface material or the surface properties are really that crust. So it might be kind of crunchier or have a different flavor. And if we were to have that same amount of bulk bread, but instead in a crouton format, we have a lot more surface area. So we get a lot more of that crunch or that flavor we associate with crust, right? So we're really distilling down the surface property of bread when we chop it up into that crouton format. And similarly, for kind of bulk materials, when we have them in the nano format, we get a lot more of those surface properties per amount of material. And so this has really already been leveraged a lot in the technology space, especially electronics. When we think about energy transfer through a bulk material, that can be really tedious to transfer energy, heat, electrons, or anything through a bulk, especially metal, which is usually what this would involve. And so instead, if we utilize nanomaterials instead of transferring through the bulk material, we can kind of pass things along the surfaces of the nanoparticles, which is a bit more of an efficient way of kind of transferring energy. And so that's what allows us to do a lot of kind of the quantum computing we think about, or even kind of our phones today, kind of having pocket-sized computers, is due to nanotechnology that's been applied in that space. And so really what we're trying to do is leverage these unique emergent properties of nanomaterials toward biological applications. And now I've used the word nanoparticle or nanomaterial a lot already, but I did just want to kind of explain that this is, of course, a really big umbrella term. So nanoparticles aren't any one material or any one shape. Nanoparticles can be made of inorganics, so those metals I talked about, like gold, kind of we've seen in tech, but also are utilized in nanomedicine. We've also seen a lot more of kind of soft nanoparticles or polymer or lipid-based systems. And even within these categories, there's dozens of subcategories and kind of an endless list of possibilities. So a lot of my job as a kind of nanomedicine researcher is to either utilize these existing materials or synthesize new materials in these spaces. But the overall goal is just to design the right tool for the job. And the job we have is the better delivery of medicines. And so, really, I think a lot about how these material properties can affect the delivery of medicines within the human body. And really, kind of what nanoparticles are able to do for drug delivery, I think is best understood if we think of an example. So, for example, for a cancer patient, as that's really what I work the most on, if they were to have a tumor and we're delivering something like a chemotherapy, which can be really toxic, if we just deliver it into a patient and it goes everywhere, it can help eliminate some of that tumor or deliver medicine at that disease site, but it's also going to go a lot of other places and have impacts outside of just the kind of function of your therapy or eliminating those cells. And so, if instead we utilize a nanoparticle platform that can confer things like targeting or help localize that drug to our particular disease tissue, we can achieve that localized impact to help avoid side effects, improve safety and efficacy. And we can also think about nanoparticles as a way to improve the solubility or bioavailability of drugs. So, some molecules that might have been impossible to deliver without some sort of aid to allow them to dissolve in aqueous solutions. It can also help with protecting fragile therapies. So, there are some drugs that just might not survive circulation to reach their target tissue. And so, by utilizing a nanoparticle, we make it more feasible to use those drugs. And so that's kind of a lot of different factors we're considering at once, or a lot of things that these nanoparticles could do to help improve drug delivery. And so a lot of what we think about is how we're going to design the right tool for the job. And so for every application space, we kind of think through a lot of the same questions. We think about administration route. So how do we want to deliver this medicine to a patient? Do we want it to be injected, applied topically on the skin, maybe even inhaled? How is it going to enter the body? We then think a lot about biodistribution or where we want the medicine to go. So if it should go to a specific organ, or if we want it to only go to diseased tissue versus healthy tissue. And then we also think a lot about the idea of immunogenicity or biocompatibility, which is really just how are these nanoparticles interacting with our biological system? Are they going to kind of set off alarms where immune cells are identifying this as a toxin or a threat? Are they going to be well tolerated by the body? And so I'm just going to give kind of a brief example in each of these categories of ways we can change nanoparticle design to change kind of how we approach each of these questions. And so, for example, for administration route, if we know that, say, we want a nanoparticle to be inhaled because we want it to go to the lungs, we know that within our airways, there's a lot of biological barriers, including mucus, which is kind of just a thick coating you have all along your airways and in your lungs. And so, through past research, we know that the size of our nanoparticles and its surface charge can really affect the ability of this material to get through that mucus layer. So, for example, we'd want negatively or neutrally charged nanoparticles that are below a certain size. And so, when we're picking a material or designing a nano platform, those are the parameters we really look to tune for that sort of delivery. If we were to consider biodistribution or where we want our nanoparticles to go, if we know we want them to collect, say, at a tumor site, we could look at a number of different things, their size, shape, charge, and surface coatings that allow them to accumulate in the tumor. So, kind of each one of these properties does ultimately affect that accumulation in some way, but a major one is we know that kind of the neutrally charged nanoparticles shown in green and the small rods and spheres tend to accumulate best in the tumor. So, we'll try to use those shapes for that sort of drug delivery. And then lastly, on the immunogenicity front, or kind of that biocompatibility or safety element, what we're looking for is to really avoid interaction of our nanoparticle systems with immune cells, because this can kind of set off the alarms within the body that there's a foreign agent present. And so what we like to do is use surface charge as a way to mediate those interactions. but also polymeric surface coatings. So we'll attach a polymer that acts as kind of a shield or like an invisibility cloak for our nanoparticles that helps avoid any recognition from the immune system. And I wanted to specifically point out one polymer in particular that we use. We utilize across a lot of different nanoparticle platforms, we utilize polyethylene glycol or PEG. And I introduced this because it's used so broadly in the nanomedicine space. We use this to coat nanoparticles at different densities and in different ways to increase nanoparticle stability, avoid that immune recognition, and alter cell interactions. But other reason I really wanted to highlight PEG here is just because I think when we talk about nanomaterials, it can sound like they're a whole subclass of materials that are separate from what we interact with in our day-to-day lives. But PEG is something most of us see every day. It's in our Tide Pods, it's in stool softeners, baby wipes, even tooth whitening strips. This is a polymer that we're utilizing, maybe not at the nanoscale, but in a number of household spaces. So I think something I wanted to emphasize was just these nanomaterials are simply normal materials we have in everyday life, just shrunk down at a smaller scale. And so I'll kind of leave that as just the broad introduction to nanomedicines, but just a few takeaway points being that really the efforts here are to develop small tools that'll have big therapeutic impacts. And we're utilizing this nanoscale not only to give us unique material properties, but also to let us connect with biological systems. And ultimately, ongoing nanomedicine research is in the efforts of engineering platforms to deliver therapies with specificity, safety, and control. And I'll turn it over to Christine to cover more on kind of the lesson planning around this as well and how to share this effectively in a classroom.
Maggie, that was amazing. Again, I'm very lucky to work with the Marble Center and the Koch Institute, but it really is our researchers who really help bring this research to life. Um, so, I'm going to give you a Zoom flashback of, wait, let me get my slides ready. We all miss those, right? Okay. Um, so, uh, just going to talk about, it's probably what I'm going to talk about is nothing new to all of you awesome educators, but just going to come at it from my perspective. So when we're talking about things like cancer research or nanomedicine, I do my best to make this content accessible. I'll tell you a little bit about my background and what we do, in particularly in the Koch Institute. Again, if there's any questions that come up, please feel free to reach out to me. And I'll also make sure we give this email address at the end. So, quick about me, our educational outreach, the 5 E's of science, and I want to give people some resources that, if anything else, I have, you will have one more tool in your toolbox. So, kind of explains how long ago I left teaching. 3 years ago during COVID, if you remember the wonderful Bitmojis, I'm still in that era. But I taught middle school, sixth, seventh, eighth grade for 7 years in the Cambridge Public School District, and the population I taught were what we call sheltered English immersion in other parts of the country known as ELL learners. That gave me the specific background that I can scaffold and make sure that different scientific concepts, from talking about atoms and molecules to talking about heredity and genetics, we try to make it accessible to students at all levels of science content background knowledge. And just any learner with any background knowledge will always be beneficial with more visuals and more kinesthetic tools. My role here currently as educational outreach, I've had for 3 years. And just to kind of talk about my role, part of our mission is that we want to tell our greater community about the cancer research that we're doing, because it is great to have these breakthroughs in science, but it really does help give the population hope that things are getting better. So just to talk about educational outreach at the Koch Institute, um, so some of the programs that we teach: investigation, cancer research, and genetic risk program that we're going to open up in April. Basically, we concentrate on a complex topic. Investigation program talks about uh, target drug delivery as delivered by nanoparticles, and I'll speak a little bit more about that. our cancer research program, we talk about tumor heterogeneity and how cancer cells become chemotherapy resistant. And with the genetic risk program, we're going to talk about how does having a higher genetic risk of a mutation affect certain cancers and early detection. Yeah. Our investigation program, I wanted to concentrate on. Um, Basically, in our program, we make sodium alginate. We make like a little boba ball, but it's a great introduction to middle school and high schoolers in particular to talk about nanoparticles, medicine on a nanoscale, in particular, chemotherapy. How does it work? It is a chemical that is sought out due to its toxicity, in particular, its ability to kill any fast-dividing cell. So it's not particular to cancer. It could also affect our healthy cells. Well, what are we doing about that? We want to create a transporter to make sure that it is only attacking the cancer cells. So, kind of how Maggie already mentioned, like we use some liposomes, and just using positive and negative charges, we can create finite medicine to kind of help with cancer treatment. Kind of like the gobstopper. If any of you remember, I'm not sure they still make it. Our middle schoolers tell us that yes, they still do, which is concerning, but meh. Um, all of that to say, like, we talk about nano carriers and make it really accessible. Um, Paula Hammond is one of the leading researchers in nanoparticles, and we kind of just talk about how I read to them that Professor Hammond is a pioneer in nanotechnology research with a program that extends from basic science to transitional research in medicine and energy. She's introduced new approaches to design development of complex drug delivery systems for cancer treatment and non-invasive imaging. And that's important for them to know because, like, one, these new insights and new developments are affecting people in every day and especially in their treatment. And it also speaks to like not only that we know that chemotherapy is in the center of the medicine, but that in between the layers, we can also put nucleic acids in there that can turn off the cancerous gene. So it's something very simple. Kids really enjoy making the sodium alginate to make a little boba ball. But we talk about it's just like new innovations, new thoughts, and why it's so great to collaborate together to create things that hadn't previously existed, but solve everyday problems. That said, uh, this is where I say I'm like, this is nothing new to anybody. When designing our program, in particular, lesson planning for our three lessons, I always consider the five E's of science. In every program that I run, we engage. I want to give them an introduction to the Koch Institute, what is a cancer research facility, who works here, what are our buckets of concentration, what do we do? I always give them an opportunity, the students, to explore. We usually have an interactive activity, very kinesthetic, very hands-on to help create understanding behind research and kind of to demystify. I think sometimes students, just like right in STEM in particular, they're like, I'm not good at this. I'm not going to continue with it. This is not for me. They find themselves having fun and at least gathering a little bit of understanding is something that will apply to the real world life. And so, and I've even had adults in the program be like, oh my God, I understood that. And I'm like, yes. So that's kind of our goal to that people understand the research that we do and to go into explain and elaborate. Students usually get to prep their questions ahead of time, and then they have 30 minutes with a researcher, just like Maggie, many other volunteers that I have at the Koch Institute. I'm very grateful that they explain and give back to the community. And we also do a tour of the lab floor so they get to see our facilities and kind of what we do. Last but not least, evaluate at the end of the program, usually that lasts about 2 hours. Um, I do a Kahoot, and just like any teacher knows with their exit ticket, I want to address any misconceptions that students may have as they're coming out. I want them to understand a little bit more about chemotherapy. I want them to understand about cancer cells. I want to make sure that they know At the Koch Institute, we concentrate on putting life scientists together with engineers and kind of creating this Avengers initiative. And that collaboration is key to any innovation in technology and treatment of this disease. Um, so we do definitely do that review. Do do do. And then I do want to give some resources. I will show these. I have the links, and I will make sure Maria Fernanda and Mario get these. Um, just one of the things that people say, like, "Oh my God, I know you, so you can get my students in." I teach students programs, but there's wonderful opportunities for high schoolers in the Boston area. We do make sure that these opportunities are welcome to students from all over the country, whether it's our MITES program that gives them opportunity to look at college, or if they're interested in doing research. We have neighbors at the Whitehead Institute, Novartis Institute, Broad Institute that do summer research programs and mentorships where these students are paid a stipend. So I'm happy to share those to make sure everyone gets the opportunity to look at that. But not only for nanomedicine, but any research that's going on, we have the KIMIT .edu news. These are a great source of up-to-date innovations, usually written by MIT. General reader, what do you call it? Reader level is high school level, I would say like ninth to 12th grade. Understandable, already accessible. We also have KI images that we do. Behind me, I show the images that we have currently in our Koch Institute gallery. These are current images of research that's going on in life science at MIT. So if you're ever looking for images that you can use to gauge or explain, we have a whole archive of it. And I will show you kind of how it looks like. And also, we're on YouTube. I have to promote our YouTube page because I don't think we have that many followers. But at Koch Institute at MIT, we have great explanation videos of a lot of innovation resources that we have. Last but not least, I will also just show this human cell tree map. If you ever wondered the size and number of a certain cell in the human body, we have this human cell tree map that a group of scientists are actually monitoring the size, the scope, the multitude of different cells in our body. So it is a great resource to when you're teaching genetics and heredity. Um, all of that to say, that's kind of how I implement uh, higher concepts. Again, I use my resources. I'm very lucky I have an abundance of volunteers like Maggie who will explain nanoparticles to me, try to engage the 5 E's, and definitely seek wisdom from our neighbors at the other institutes that if we can't provide something, we don't have to reinvent the wheel. I talk with other outreach specialists and they kind of help um, facilitate that. But with that, I think we're ready for just informal questions.
Thank you so much, Christine and Maggie. That was awesome. That was a great crash course of nanomedicine and also some really good resources that can be used in the classroom. Who would like to start with a question? Marielle, please go ahead.
Hi, ladies. First, I'd like to say that that was such a wonderful introduction to what you do, Maggie, and also Christine, how you share it out to the rest of the world. Um, uh, so, are these nanoparticles? We've been waiting for gold nanoparticles for a really long time because I know that they can be sensitized to attach to tumors. Is any version of this actually in trial? Like any version, maybe not your version, but like, injecting nanoparticles for cancer.
Oh, absolutely. There's definitely ongoing clinical trials for a number of different materials too. For kind of the gold nanoparticles we had talked about, there's a lot on kind of photothermal therapies, which I didn't dive into too deeply here, but you can essentially, if you have a gold silica nanoparticle, you use a laser to kind of excite a particular amount of heat off of them, and then you ablate the cancer cell in that way. And so there's, I believe, still phase 2 or 3 clinical trials for that for lung cancer. But a lot of the softer particles, there's actually already There are, of course, there's been kind of nanoparticles approved for vaccines and other applications as well.
Right.
And there's been some really similar formats for kind of personalized cancer vaccines. And then kind of the most exciting ones we've seen recently, where it's kind of delivering a nucleic acid. But there's also already nanoparticles that are liposome based that deliver different chemotherapies as well. So we do have kind of some approved, more exciting ones in the pipeline, but it kind of spans the whole gambit. There's absolutely some of these in clinic, though.
Thank you. And just so that I understand, the thing that I tell my students when I talk to them about this is if they've ever known anyone who has had cancer and had to go through chemotherapy, how awful it is. And so many kids have some kind of connection to that. And that this is avoiding that because instead of systemically treating the entire organism, you're just targeting, right? Is that true? Is that
Yeah,
exactly said about
absolutely.
this?
No, that is exactly, and I think the best way to describe it, because I think a lot of people can understand what like side effects are a really good motivator for why we want the nano platforms because it can sound like it's this like, the long list of things they read on approved therapies when you have the commercials on TV. It's like, oh, side effects include. It's like the
Yeah.
shorter we can make that list, the better everyone's life is. And it's
Yes.
exactly that because we're targeting the impacts. I guess I will amend that kind of a lot of the approved therapies right now are not yet some of those targeted therapies, and that's more what we still have in the clinical pipeline, but they're definitely on the way.
All right. Well, thank you.
And that's a fun thing to always explain to students in particular. It's just like, yeah, you remember those side effects where they're like, yeah, might include death. I'm like, if you guys think of biology as subject, from 0 to 100, we're at 15% of what we know. And so one thing I explain to kids is like, think about cheese, harmless cheese. I'm like, do all of us react the same? And I'm like, no, some of us will have it every day, and some of us will die a horrible death on the toilet. And so
That's a great example.
I'm just like, and that's just cheese. So I'm like, when we consider everything that might happen, everyone is so complex, and that's why we're continuing to discover more in the subject area.
You know how to talk to preteens.
We have a question in the chat. From your experience, what are the most important and influential teaching tools that you have on hand at the Koch Institute?
For me, I would have to say my predecessors did a really good job of uh, taking tangible um, discoveries that we have, like the nanoparticles as a target drug delivery system, to talk about encapsulation that kids are familiar with, in particular, like in comparison to Tylenol, right? We have a little capsule, we take it out, you see all the little medicine inside of it. Um, but I really do try to make sure that science is something that's tangible and explains the way we know about the world. Um, in particular, when we're talking about cells, like all cancer is one cell off a mutation. And one of the things I compare it to is that if they've ever done scratch coding, all our cells have that make a copy until. When we don't have that until, you just get a horrible line of code. I am very lucky that all these scientists, especially in our galleries, take the time to kind of explain and break it down for the general public. And I just make sure I break it down a little bit more to make sure it's accessible. If there's something simple, one of the things that we do for chemotherapy resistance is that we use colorful beads to describe different cancer cells in a tumor. And one person is chemotherapy, putting the beads on the plate of death. And the other two people are looking at the plate of death and trying to put them back into the cancer cells, back into the tumor. And at the end, we count them all and reassess and do diagnosis. Um, I think anything, just like other teachers know, is just like anything that we can do to make it tactile so they can see each of the systems, um, definitely helps that exploration explanation process. And just, I do try to demystify that it's impressive to come from MIT. And what's impressive is the people and what they do here with the resources.
Thank you. There's also another comment from Tarek. Oh, he just wants to add that gold nanoparticles are also widely used in diagnostic space. For example, the pink bar you see in COVID test or other similar or diagnostic devices are gold nanoparticles. I think we've discussed that in the past too. I also wanted to point out when Maggie was talking about chemotherapy in liposomal form. Fun fact, I believe the first FDA approved nanomedicine formulation was Doxil, which is doxorubicin, the chemotherapeutic agent, into liposomal bubbles, liposomal nanoparticles. Any other questions? I'll jump in. I'm curious to hear what are the common questions that students bring up when they to MIT or when you introduce this type of material to them? Is there any common threads that you keep seeing coming up over again? Something that might spark their interest a little bit more than other topics?
Wait, who is it directed to?
Whoever wants to take it.
I was,
Because I know some questions that I get, I feel like I definitely don't have as much experience as Christine in terms of kind of number of folks I get to interact with about the material. But I do think that we get a lot of questions actually around, I think chemotherapy is the point that a lot of people understand because it's a word they've heard or seen on TV and like have seen what those side effects look like. So I will say like the concept of side effects and chemotherapy crossing with nanoparticles, we get a lot of questions about for sure. The other thing I feel like I do get a decent amount of questions about are as soon as you mention one nanoparticle, there'll be a lot of questions around kind of how is this nanoparticle different from this other one I heard about? So a lot of times comparing nanoparticles for cancer research with, say, like the vaccine nanoparticle, just because that's one that everyone's like, oh, I've gotten a nanoparticle and it was this one. So how is this nanoparticle different? So I do get asked sometimes about why one nanoparticle is picked for a vaccine versus, say, kind of a cancer treatment, or why it's we can't just use the same one for both. And I usually just try to answer that around kind of the idea of the right tool for the job, where maybe the vaccine is like a hammer that's great at getting a nail in a wall, but maybe you don't want to hammer a screw into a wall. Maybe you need a screwdriver. So my job is to make the other tools. And I feel like try to explain it kind of in that way because you don't want to, make other nanoparticles look bad because they're great at their job, but knowing that it can't be applied everywhere.
and I'll just say like to piggyback off of that, I think the greatest misconception that a lot of students ask is this like, what do we have as our options for treatment? Like radiation, chemotherapy, or immunotherapy they may have heard of. We talk about CAR T cell therapy that is successful in 60% of like blood cancers as a form of treatment, but in 40% it doesn't work. Um, and so like we kind of talk about uh, why is it very important to do personalized patient care? And when we're even doing our like diagnostic in our uh, simulation of our chemotherapy resistance, um, we also talk about what factors would go in the lifestyle of a person, the age, if they can take the treatment and different things. And so I think altogether, kids have a general curiosity of like not only what is cancer, we say it's a disease, but like what is the implications of the disease and how do we treat it in a way that is humane and effective to each patient. And kids get really interested in that aspect of it.
Thank you. Go ahead,
Christine,
Mary.
I'm just wondering, thank you. I'm wondering if you do you push into classrooms or do the kids come to you?
Um,
And
kids
and I have
usually
a follow-up question too. Go ahead.
You're fine. Usually for like our 2 hour program, the kids usually come to us. Um, and that said, I'm not against pushing into classrooms, but I think uh, as a former teacher during the pandemic age, I think that there is like something nice to be able to, when possible, of course, get to see the lab floor area, the lab space. Um, Again, I do explain to them, I'm like, who works in our building? We have postdocs, we have PhD students, we have principal investigators. And it's just like, not everyone who works here went to MIT as a scholar. So it's just like, you can work here without going to MIT. That said, you can go to MIT as well. Um,
my school work closer to you. And have you any activities that you or lesson plans or anything that you can give to teachers to work with this or just on topic of these types of this type of research?
I don't have any activities per se that because our programming is under the umbrella of MIT, like anything
Hmm.
I create program-wise is property of MIT. So there's
Okay,
certain things that I am able to share and certain things I am not. Um,
right.
that said, there are some public uh, programs for, I know, math and science that I have available, and I'm happy to send that to Maria Fernanda to share with the group because there are two tools that I'm thinking of currently, but I'd have to bring them up.
That would be great if you would be able to share, because we're always looking for new ways of putting this kind of really cool information and into classrooms so that students have an awareness of like what the options are, right?
Exactly.
And to let them know that they can do something like this to impact, a large number of people. So my
Mhm.
students who go into all of my girls want to go into being, a doctor, and because they want to help people. And I always tell them you can help one person at a time as a doctor. But if you go into research or if you go into engineering, you can help lots of people, right? With the stuff that you do and then the impact that you can have. So the stuff like this, research like this, is really important to share with students, I find.
Abs-
Yeah. So anything you can share would be great.
Absolutely. And please, I do encourage those resources that I send out for high school opportunities. Again, those are for students all over the country. Even Mite's program runs with some students in Puerto Rico as well. That kind of gives them preview to college life previous to college. They get to go to classes, be in the dorm, and they're paired up with a guidance counselor to kind of help them with that transitional phase between high school and college. And then the Broad Institute of Artists in Whitehead all have programs where like students get paid to do research in a lab and they're paired with a mentor and they do end up showing it on a national stage. And then the last resource, I believe, I can't remember, it's the US Foundation, National Science Foundation. But that also is for it states all over the United States of the different STEM programs that are available to high school students as well.
Yeah, I saw that. I just found the doc.
No, no.
You shared so many links. Trying to
I
go through
did.
all of
I
them.
did. I went link
Yeah.
crazy. I apologize.
No,
I
it's
got
but
so
it's
excited for
wonderful.
everybody.
It's really appreciated. It's really appreciated because this is the kind of stuff that um like that I want to share with my students, right? So that they can see, so that they can potentially get involved. Oh, this is wonderful. Thank you.
Thank you. Okay, there's more questions in the chat. This one's from Yara. Is computational involved in the recent research? She says, for example, recently more emphasis is announced in novelty of AI and machine learning. Is that being utilized in teaching or maybe Maggie in your research? How do you see this aspect being important or helpful? And I would like to add an extra line of it. How do you introduce it to students without being overwhelming?
Yeah, there's definitely a lot of kind of that computational and machine learning and AI incorporated into nano research currently. I would say maybe just to kind of, there's a lot of excitement around it. I will say that it's definitely in the early stage, I think, implementation in a lot of these research spaces, just because you need a lot of data in order to use those systems kind of meaningfully. And so, of course, we have a lot of kind of just gathering enough data that we can actually kind of train a system to better understand nanoparticle performance. But there's actually folks currently in the lab I work that are doing that now, and kind of generating big pools of data that can then be analyzed. So, I mean, we're not the only lab doing this, of course. We're definitely seeing that all around. Again, it's just challenging to find the right type of data to use in those systems. But there's definitely a lot of potential for kind of selecting the materials and predicting how we'll perform in vivo, which is like a really kind of, I think, hard part of cancer nanomedicine research. The other thing we're seeing a lot of, and I think maybe kind of more readily translating, is applying those systems to just cancer research in general. So understanding cancer evolution, predicting how cancer will evolve within a patient, and correctly identifying genetic vulnerabilities for patients. So a lot of like cancer genome work, I think, is maybe kind of overlapping with nano because we'll find, oh, we should target this on the cancer with our nanomedicine. So it's
Mhm.
absolutely happening. I would think just we haven't seen it fully translate or come to fruition yet, but it's definitely in the works.
Thank you. Another question from this one's from Charles. Is anyone aware of nanoparticles being developed that can deliver across the brain-blood barrier?
You
Oh, I
should
think I
Yeah.
lost you
definitely
guys.
speak to
Um,
that.
Yeah, absolutely. There's kind of a few approaches within the Hammond lab. We actually have kind of a polymer system that was able to achieve crossing of the blood-brain barrier. It's just something you always want to do with a lot of intention, because of course most things we don't want going to the brain. But there was essentially kind of a polymer identified that was able to interact with receptors on the blood-brain barrier and kind of achieve uptake through one cell and then kind of excrete it out the other side of the cell into the brain. And that is what is utilized for some glioblastoma treatments, so for brain cancer therapies. But there's a lot of approaches to that. That's just within our lab, but there's a lot of antibody-based kind of strategies with our nanoparticles to achieve that.
So cool. So cool. Um, the next question, from Jose Julian. Do you know any virtual laboratory to teach nanoscience, like a page to simulate the synthesis or drug delivery? He's from Ecuador, and he says we have this gap and we cannot access state-of-the-art instruments in our labs.
Oh, he's from Ecuador. My family's from Ecuador too. Hola, Julia. So, talking with that. So originally I was going to share with you all, but I don't think it's there anymore. There was a Department of Nanomedicine that had a vaccine game, that kind of like talked about, um, let me see if I can get it in the chat, but it's not there anymore. That talked about nanomedicine kind of going in, and
Hmm.
it's a way to show, how nanomedicine worked. There is another interactive game, but I have not verified how well it works. No, this one doesn't work as well. I would think for nanomedicine, there are instructional videos. For example, if I'm able to share what we have from the Image Awards, um, pardon me, and I apologize for all the back and forth. Um, Let me turn off my alarm. Uh, so for example, when people were talking about like, do we have anything to go through the brain-blood barrier. This actually from Kimberly Bennett, also part of Paula Hammond's lab, created this microchip that's very small. And basically you can put brain tissue on it and it replicates the brain-blood barrier. So we test nanoparticles to see its efficiency. But like, for example, this is a video I currently have up, but there's also videos that we have with more student-friendly language that kind of explain with slides and vivid detail from the researcher. Um, so it's not so much of simulation, but it's something that can kind of show the understanding of like how we design the nanoparticle in order to fit different areas of the body. Um, and so and I will make sure right now the one that we have for with more student-friendly language isn't uploaded yet, but there are a couple of them that we do have available.
Thank you. I would also like to share there's something called NanoHub. I'm gonna put that on the chat. And there's a wealth of like models and simulations and teaching activities and tools that you can use. Um, I'm pulling it up right now, and they do have pharmaceutical formulations, an intro to drug discovery and delivery. So those might be really useful to try simulations in the classroom. And those resources, I believe, are completely free. Ooh, lots of stuff in the chat. Oh, we continue the discussion on AI. Okay. Yara has another question. Have your team tried experimenting with topical formulations of nanoparticles? Do you know if the easiest and safest type of pharmaceutical formulations to be processed almost instantly using topical lipid nanoparticles? Oh, that's okay. Just sharing ideas from my experience and looking to see if you agree with me. Yara, would you like to expand on that?
Hi, can you hear me?
Yes, hi.
Hi, how you doing? Happy to see you, Dr. Maria, and thank you everyone for the beautiful presentation. Really, this is a topic that is a favorite of mine. So I am a pharmacist and I'm doing my PhD right now, and I've had some experience with in the lab doing topical nano particles, specifically lipid nanoparticles. So this is something that, it's an idea I wanted to share and also see if anyone has experimented with this before. I think it's very interesting because this is something that could be easily formulated, and if you have kids or high schoolers, they could walk out of the lab with, having particles with them, which is really cool as a cream or as a topical lotion. Some really simple formulas could be done in the lab that are 100% tested. For example, if you're talking about a lab that already has the processes conducted and tested and approved already. So in pharmacy, this is something we also do in the compounding pharmacy. So they are very simple formulas that involve mixing. And for the nanoparticle part, it involves the size reduction type of mechanism. So this is just a simple idea. Like, it's very interesting that you could walk out with some nanoparticles in your hand. This is just something to help the kids visualize. This is always something helpful in the learning process. So I just want to share this example from my experience and see if anyone have thought of it or if you have already labs for topical formulations. And yep, looking forward to see if you have an idea on this or a comment. Thank you.
Thank you for sharing, Yara. Any experience with topical formulations from anyone else? Yeah. Well, thank you for sharing, Yara. Any other questions from the group?
Hi, can you hear me?
Yes, hi.
Hi, my name is Aldo, and I'm here in Phoenix, Arizona. So, first of all, congratulations on the amazing work that you're doing there in the lab, as well with the outreach program out of MIT. So, I have a few questions that I'm not going to monopolize all the time. The first one is when you do have high schoolers to come in and do some sort of internship in the summer, how long is that approximately?
Um,
Uh, Maggie, anyone?
Oh, I was going to speak to that. So the Koch Institute and Marble Center don't necessarily do any internships, but for the Novartis, Whitehead, and Broad Institute, those can be anywhere between like 4 to 6 weeks. I would say definitely check each program. I do say I know Broad and the Raygon Institute, those applications are probably due sooner than later. So if I don't know if it's something that you could prep students for next year's to kind of see the application. But yeah, those are usually 4 to 6 weeks, and then they get a chance to think of their own scientific question paired with a mentor, work in the lab, and then they have to carry out the presentation, kind of science fair-esque, record their results, and talk about it on a more formal stage and present it in a very similar way to how our scientists present their discoveries.
So it's a pretty well organized, rigorous type of um, but again, that's not an internship, it's some sort of research.
Yeah,
Is
it's for research, and I would say like, for example, the Broad program has been doing outreach for 20 years, so they have one of the most fine-tuned um, programs in the country. But unlike that said, I think um, all these institute are no more than 2 blocks from each other. And so, like, there is a lot of opportunities that we try to give for students all over the country.
And who's in the selection committee? How do they get chosen? Is it based on their current desire course of studies? Is it something that they can prep for and to have a better chance to get admitted to it? Or how do you go about selecting those few students?
Oh, I am not necessarily in the process, but I have talked to the outreach coordinator at the Broad. Usually there's a selection committee that kind of look based on opportunity, availability, um, and then looking at fine-tune factors if there's like um, usually they look at students with financial need and who haven't had opportunities. They don't want that being a problem. But that said, I think that they do curate their applications because they do get a lot. So I'm not really too sure other than I know that they have a committee, they have look for us, and they try to make it as equitable as possible.
And do they get to choose the area they're going to be working under, or do they get
Yes.
assigned to someone?
They based on the subject that they want to kind of get their answer in, they usually get paired up with a mentor. And they're lucky, especially in the Broad program, that they have like mentors sign up every year. So they have a lot of um, repeated mentors. And in particular, it's nice because then they have experience of how to work with the students. But yeah, for the most part, I'm like, they do pair them based on interest in order to get the best results.
So since you mentioned, they do look into, financial needs,
Mhm.
I suppose that as a minimum, they have to come to your labs with a personal laptop so they can do some writing, some note taking, some presentations, or do you provide something for them? Do you know of that?
I know that they do at least, um, they kind of have like a shared community space. Like, even if they wouldn't have their own personalized laptop, they do have Chromebooks. They do lend out for students who don't have it. So I think that they do have like accessibility to use the Chromebooks in order to um, be able to carry on with their research.
And do you know the start and the end date? Is it because out here we're done with school end of May and we get back to school sometimes late July. So when is the programs that you know of?
I know that they have different starts and end dates. It would definitely, like, I would double check the websites because I don't want to say off the top of my head and we're being recorded and I might be wrong. So I would definitely say, definitely check the websites
Yeah.
just in case because I don't think that each program has the same start and end date.
Okay. One last question, although I've taken too much time, is to Maria Fernanda. So now, as I stated before, I'm new in this space. So how do I continue getting updates of your next meetings and now that I'm registered, would I get some follow-up emails, some follow-up anything?
Well, thanks for asking. So everyone that registered today will get a summary of just a little summary of what was discussed today with all the links that we shared and all the links to the resource page. Um, I try not to put anyone on list servers that they don't want, but I do keep a list of everyone that has registered, and I make sure that they get the invitation for every single NanoEd forum that we have. We meet 4 times a year, so you don't get a lot of emails from me. Um, and you're always welcome to email us if you have any questions or if you're looking for specific resources for your students. And with that,
Okay.
we're at the top of the hour.
Mhm.
Thank you so much again to our speakers. Thank you, Maggie. Thank you, Christine. This was a lovely presentation. I will make sure everyone gets all the resources you shared. And see you all next time. Bye bye.
Thank you so much. Bye.
Thanks, guys, so much.